I'm Katie Vermeersch, and I have the pleasure this morning of introducing my advisor, Dr. Mark Styczynski. First, I'll start off with a little bit of background on Mark. He got his bachelor's in chemical engineering at Notre Dame, and then he left the cornfields of Indiana to venture out east to MIT, where he completed his Ph.D. with Dr. Stephanopoulos, and he received that in chemical engineering as well. The next hop wasn't quite as far. It was only over to the Broad Institute at MIT, where he did his postdoctoral studies with Dr. Ragaz until 2009, when he joined Georgia Tech as an assistant professor. Currently, his research focuses on the dynamics and regulation of metabolism and joins together both experimental and computational research. The experimental part gathers new metabolomics data from many different types of cells, such as cancer, stem cells, and yeast. And then the computational arm involves the development of new bioinformatic technologies in order to be able to properly handle and process the data gathered by the experimental people. Now, without any further ado, I'd like to hand it off to Mark, who will more in-depthly talk about his research.
On. Okay. Are we good? Yes? Can we hear my? All right. Excellent. All right, well, thank you very much, Katie, for the introduction. Katie is actually our senior graduate student in the lab, and she produced a whole bunch of the data that I'll be talking about today. And today I'll try to give you a kind of vague overview of some of the things that we do in the lab. There are other things that we do in the lab. We've sort of got our hands in a lot of different pots, but we do a little bit of synthetic biology as well, and I won't get into any of that today, but today I'll talk about some of our work about uncovering metabolic regulation dynamics. And so just to give you an overview, I'll sort of start out talking about yeast, because that's where sort of my intellectual motivation, our lab's motivation started off. And then specifically I'll talk about one of our projects, which is trying to identify novel metabolic regulatory interactions in yeast, and then finally I'll talk a little bit about our work in ovarian cancer. So first, before we go anywhere, I want to make sure that we're all on the same page, because I talk in omics talk, and so I want to make sure that we all know what the words that I say mean. So first of all, metabolism. By that, I mean the set of processes that take nutrients and turn them into energy and the building blocks for new cells. So we're taking things in and we're turning them into new cells. We're using all these different pathways in order to make new cells and all the energy that they need to survive. Next, metabolite. You can have a ton of different definitions here, but what I'm specifically interested in are small molecule intermediates of metabolism, typically less than 500 Daltons, because that's what we can easily detect on our mass spec. And also, I'm most typically interested in polar molecules as opposed to lipids. So you'll see a lot of focus on that, things like glycolysis and the TCA cycle. And finally, metabolomics. Metabolomics is the metabolite and metabolism analog of genomics. What we're trying to do is we're trying to measure as frequently as possible, as much as possible of the metabolome as possible. So we want to measure as many metabolites as we can in a given run. So those are preliminary definitions, and hopefully that's enough for us to get by. One of our main techniques that we use in order to analyze these metabolites and do our metabolomics work is a chromatography separation step coupled to mass spectrometry. So what you see here is we have our nice auto sampler that helps to keep our graduate students from just standing at the GCMS and injecting every 40 minutes. And then we separate, and we actually have a 2D gas chromatograph. It actually gives us orthogonal separation in 2 different dimensions, and there'll be a picture or 2 that shows you the kind of data that we can generate, but we actually wind up with really sensitive detection of metabolites because we get exquisite separation based on being able to separate in 2 orthogonal dimensions, say boiling point and then polarity. And then we run that through our mass spec, and then we hopefully get readouts when the mass spec is working. And when it's not working, then we just try to put everything back together again. So I'll start off, before I go into a lot of depth about what our lab does, I want to make sure you understand why uncovering metabolic dynamics and regulation is actually important. Because you might say, based on, say, one of these previous slides, well, if we know all of this about metabolism, what's the point of going on and trying to study it in a lot greater detail? And so I'd like to give you a good understanding of that, because when we look at metabolism, and you have this picture here from the KEGG pathway database, when you look at metabolism, we know what the individual nodes are. So if we treat each of these dots as a metabolite, and each of these lines connecting the dots an edge, if we treat each of these edges as a reaction, this overall is extremely well known. If you look in E. coli, if you look in yeast, if you look in humans, even if you look in more exotic extremophiles, these things are all fairly well characterized. Not perfect, but pretty well. However, when you look at what we can do with these pathways, when you think about what a yeast does versus what a human does with these pathways, you can get extremely diverse macroscopic phenotypes. And specifically, one thing that is of interest to some graduate students is things like the Crabtree effect, the ability of yeast and other organisms to actually produce a whole bunch of alcohol even in the presence of oxygen. So we have one set metabolic pathway, but we can see on a macroscopic level many different phenotypes. So a pretty useful approach in biology is to use comparative analysis. And if you take, say, a list of 14 yeast species, so the details here are not critical, but this is 14 species spanning over 300 million years of evolution. So these are extremely diverse species, although the guys up here are actually pretty closely related, as you can tell from the lengths of the branches in the dendrogram here. Just to give you a reference, chimps diverged from gorillas around 8 million years ago, and from mice around 100 million years ago. So from here to here is pretty divergent, although there's a lot of closely related guys up there. So we should be able to see, if you think of a comparative genomics approach, people would take genome sequences and start comparing on a gene-by-gene basis what the similarities are, what the differences are, and try to learn something from that. We're going to take that approach on both a transcriptional and a metabolic level to try to understand what's going on across evolution. This is some of my old work that I'm talking about here. And so a very fundamental, baseline, simple experiment is to grow up some yeast in a limited amount of glucose, and eventually they run out of glucose and they have to undergo what we call a dioxic shift. They shift from growing on the glucose to growing on something else, typically ethanol that they've already produced. And if you then want to compare two different species, say species A and species B, you'll notice that they're going to have a slightly different growth curve. And so if you want to actually compare things in a physiologically analogous context, all you need to do is identify, like, oh, well, here is where my curve takes a bend in the blue. Here's where my curve takes a bend in the red. And so I'm going to call that a dioxic shift. And I'm going to compare this time point to this time point in blue to this time point in red. And I can do that for a number of different time points. And what this is going to give me is the ability to then look across species, getting rid of any artifacts based on growth curves, and actually see what's going on in all the different species. So if we look at gene expression, we do a whole bunch of microarray experiments, and we actually do it for 6 different time points as we exhaust glucose. What you see here is that gene expression recapitulates the phylogeny. Now, what do I mean by this? What I mean by this is that up here you see a lot of red, which means a lot of similarity in the expression profiles. That's because these guys up here are all extremely closely related. As you move further down the phylogenetic tree, what you see is that the colors get colder and colder. We go to yellow and then eventually to blue. We're going down in correlation. And you can also see this within subclades as well. As you go further away, you get more blue. You get more blue over here. So what we see is that how close you are in terms of your phylogenetic distance, your evolutionary relationship, affects how similar your transcriptional profile is. And we could put this slightly more quantitatively and look at any individual time point and compare the branch length, the phylogenetic distance for any pair of species, to the correlation of their transcriptional profile. And we have a statistically significant correlation between them. And if you look at every single individual time point, we always have a statistically significant correlation between phylogenetic distance and how similar their transcriptional profiles are. And I think on a fundamental biological level, this makes a lot of sense. There are small stepwise changes in evolution as you are looking from one species to another, and so you might expect that there are going to be small stepwise changes in a linear fashion in terms of the transcriptional profiles. So we weren't surprised when we saw this. What we were surprised about was this. What you see here is nothing like what we saw the last time. There's no big patch of red. There's no continuous going from red to yellow to blue, because we can go from red to yellow to blue and then back to yellow. And then sometimes these guys have no similarity to anything. And then these guys go from red to blue back to yellow. So what we're seeing here, because this is our metabolite data, this is not transcriptional profiles anymore, this is metabolite profiles, that these show no clear relationship. And if we want to be quantitative again, we can go back and try to get the same correlations, and we see some just barely statistically significant correlations in lag and plateau, and there's some biological reasons for that I can get into if somebody's interested. But otherwise, when we look here in essentially the key parts of our physiological change, there is no correlation between phylogenetic distance and metabolite profiles. All right, so expression profiles are correlated to evolution, but metabolite profiles are not correlated to evolution. This makes us ask, what's going on? Where does this phenotypic diversity come from? Because if you think in terms of, say, the dogma of molecular biology, people will think that your genes control the creation of the RNA, and the RNA controls the creation of the protein, which controls the creation of metabolites. And you would think that stepwise changes in transcription would likely manifest in somewhat stepwise changes in metabolism. But we don't see that. What we actually see is conserved metabolism, because all of those yeast species that I showed you, they actually have over 95% the same metabolic genes. So we're talking about TCA cycle, glycolysis, amino acid biosynthesis. All these pathways all look almost identical across all those yeast species. So we have conserved metabolic pathways and continuously evolved transcription, which we said we think makes sense. We take those together, and all of a sudden we have discrete phenotypes. We have two species that are the most closely related acting completely different. And then we have species that are very divergent evolutionarily that are acting rather similar. And we want to understand why, because there is no good answer for why right now. And that's one of the things that we're interested in understanding is what actually controls metabolism beyond transcription. We have a bunch of ideas, we have suggestions from the literature and previous basic research, but people don't know this on a systems level scale. And that's where we come in, because we're interested in the omics, we're interested in understanding everything on a systems level. We want to look at the whole cell, all of metabolism, as much as possible. So that brings us to the first project that I want to tell you about, which is identifying regulatory interactions in yeast. So beyond, say, transcriptional control, it's pretty well documented that you can have something called, allosteric control. So allosteric control is when you have a molecule that is not necessarily a substrate from your protein that binds to that substrate. And when it binds to that substrate, it changes the shape, the conformation of the protein. So you see that a previously accessible active site, which we can represent with our star, might now become inaccessible once this metabolite binds to it. So this is an allosteric change. This is really well documented. Some of the most fundamental molecular biology transcriptional cascades are mediated by allosteric interactions. And if we want to look about how much we know about allosteric interactions, well, first you have in E. coli, there's been a little bit of focused characterization. So if you actually go to EcoPsych, you'll find that there is some knowledge about what's going on where in terms of allosteric interactions, but it's pretty restricted. And we also know that not only do we not know tons about it, but we also have found allosteric interactions to be important even in higher eukaryotes. So if you want to look at pyruvate kinase, which some people are considering to be an oncogene, because pyruvate kinase has multiple isoforms. And in cancer, it's been shown that pyruvate kinase, the isoform that is being expressed, switches substantially from PKM1 to PKM2. And that PKM2 actually has a different allosteric regulatory profile. It is regulated by a different set of metabolites than PKM1. And that actually impacts cancer metabolism and how quickly the cancer cells can grow. So we know that it can be important in E. coli, we know that it can be important in humans, and yet at the same time, we don't know it on a global basis. If you want to look at glycolysis and the TCA cycle, these are some examples where allosteric regulatory interactions are extremely well documented. You can go into your Brenda database, you can open your Voet biochemistry book or Leninger or whoever you prefer. You can go into your book and you can find a bunch of different allosteric interactions that involve glycolysis and the TCA cycle, hexkinase, things like that. But this, if you remember our picture from before, this is not all of metabolism. This is more like all of metabolism. And so we're studying a very little portion, or we have studied previously a very little portion as a community, whereas we haven't looked at everything else that can be going on. We have no idea if there's any crosstalk. We have no idea what is going on in these cells. Moreover, specifically talking about that crosstalk, we've only investigated certain candidate metabolite regulators. What I mean by that is this. If somebody is looking for a regulator in, say, tryptophan biosynthesis, they say, well, I know, based on my knowledge of biochemistry and biology, there could be some sort of feedback inhibition by the end product of my pathway. So let me look at all the products in my pathway and see if any of them regulate this specific enzyme that I'm interested in my pathway. So that's one approach that people take. However, unfortunately, that's not the only thing that can actually regulate an enzyme in that pathway. And if you look at some of the, even in the TCA cycle, you can see regulation, even in glycolysis, you can see regulation of an upper glycolysis enzyme by a metabolite that's down in the middle of the TCA cycle. So we're talking 13 reactions away. There are so many things that go on in between those two metabolites, and yet there's crosstalk, there's regulatory crosstalk between these here. So we want to start looking beyond just the simple expected feedback regulation. We want to look at the entire set of metabolism. And for those of you who are paying attention or had subscriptions to Nature in, say, the years 2000 to 2003 or 2004, about every single journal article you could see one of these little pictures, which is just a protein-protein interaction network, which even back then, in my humble opinion, meant about as much as it does now as you're looking at it. It looks like a pretty graph, but it's hard to really infer what's going on here. What I'm saying here is that people spent years and got tons of great publications and great scientific knowledge looking for protein-protein interactions, and it was super exciting. But nobody's ever done the same thing for metabolite-protein interactions. And we have evidence that metabolite-protein interactions can have a fundamental effect both on transcriptional profiles and on metabolism en masse. So this is our motivation for saying, let's go beyond the simple things. Let's go beyond the basic biochemistry that people have been doing for the past 10, 20, 30, whoever knows how many years, and let's look systematically for what regulation might be going on in these cells. So we use two approaches to try and do this. The first of these is mass spectrometry. And so I told you before that we'd have a couple of pictures. This is what a 2D gas chromatogram looks like. If you're used to a traditional gas chromatogram, if you just take this and condense it into one dimension along this dimension, you'll just see your standard peaks. But if we add a second dimension to it, now all of a sudden, this is still our total ion chromatogram. We still have full mass spectra at every single point in this. And this gives us the ability to resolve things that might have co-eluted in this first dimension, and they won't co-elute in the second dimension. So our 2D gas chromatography gives us two orthogonal separations. I've talked about that before. We can measure hundreds of metabolites pretty sensitively. And we also have a pretty good dynamic range, 4 to 5 decades. And so this is our approach to metabolomics. So the approach that we've taken specifically in looking at regulatory interactions in yeast is to say, well, okay, let's first take our protein, and whether we want to express it in E. coli or express it in yeast, we've actually changed over to using yeast, so I apologize for that not being right. If we take our protein and we express it, and we have a tag on it so that we can be able to isolate and purify it, and then we take that protein and then we add a bunch of metabolites to it. We add a complex metabolite mixture that's defined so that we know what's in it. And then we see what sticks when we purify our protein out. And then we elute, we get off everything that's actually on there. And so then this eluent is then everything that was binding to our protein here. These are candidate regulators. And then we use GC-GCMS to characterize and identify, oh, I think this is tyrosine. Oh, I think this is glucose 6-phosphate, and so that we know specifically what is binding to each protein. And we can do this in parallel for a number of different proteins. We're also sort of long-term interested in taking an in vivo approach, where instead of using a defined in vitro set of metabolites, we would actually instead just pull everything directly out of an enriched cell broth, but I won't talk about that today. So for proof of principle and some of the basic things that we've been doing, we've shown that we can, in fact, do this. So if we take ARO7, which is involved in the aromatic amino acid biosynthesis pathway, this is actually known to be regulated by tyrosine and tryptophan. And this is one of those things I talked about before, basic metabolites within the pathway that we expect to possibly have some sort of feedback regulation within a discrete pathway. And so we did our experiments, and we were able to detect binding between tyrosine and ARO7, between tryptophan and ARO7, but we weren't able to detect binding for a negative control like valine. So what we see here is that the proof of principle works. We can start doing this en masse in parallel for tons of different metabolites. For any given analytical technique, though, there's always going to be some shortcomings, and so we actually wanted to take a two-pronged approach to try and do this analysis. So we, in addition to using gas chromatography, in parallel, we use small molecule microarrays to probe for these metabolite-protein interactions. So it's small molecule microarray. So you have a microscope slide, and it has about 50 micron scale spots of different metabolites. So each of these spots is then going to be a different metabolite that you put onto an ordered array. And you know where each spot is, so for each spot you know what its chemical identity is. And so we can custom print hundreds to thousands of these analytes onto a microarray and then begin to probe with a protein. Now, interestingly enough, small molecule microarrays have been around 10, 15 years. There's a small community of 10 to 20 groups that actually actively use them. But they've never been used for what we propose, which is trying to understand endogenous regulation. They've had some amazing, great uses in chemical biology, where people use combinatorial chemistry and put tons of different combinatorial chemistry compounds that aren't native compounds, and they look, say, what could be a drug for this specific protein target. What we're trying to do is instead put, I don't want to say real things, but real biologically relevant intracellular analytes onto this array and then see what they stick to so that we can learn on a system scale about what's going on in terms of regulating the cell. So this hasn't been done before. And the approach is again similar. You're going to take your E. coli or your yeast and generate some tagged protein. And it doesn't actually necessarily have to have the fluorescent tag off the bat. We've actually been taking an approach that doesn't use the fluorescent tag and then doing a secondary hive to tag it, because it actually gets a little complicated to put that tag on there to begin with. And then in parallel, you take all the analytes that you're interested in and put them onto your small molecule microarray, which we call an endogenous metabolite microarray. And then you put them together, and what do you got? You have hopefully a protein that binds to only a certain set of spots on your array. And then you can throw it into your array scanner, scan for fluorescence, and you'll hopefully see some positive controls like this little green star that we have there. And then you'll hopefully also see some red dots, which is the analytes that your protein of interest has bound to. And now, since you know which analyte that spot represents, you can then go and pursue and try and figure out what's going on. So this is something that we hadn't done before when I got here 3 years ago. And so we dove in, and we've actually been able to import the technology with a little bit of encouragement from one of the leaders in the field. And we now have functioning small molecule microarrays. So this is great, because we went from zero, we went from a broken donated machine to real interesting good data. So we're pretty excited about this. And what you see in this microscope slide here, you actually see it in triplicate, and actually there's another one printed down there, is the green dots. These are fluorescent molecules that are directly printed on the slide, so these are positive controls. These are things that we should always be able to see. And then we see our red dots. And this was a non-fluorescent molecule that was printed onto the slide, and then we hybridized a fluorescently labeled protein. In this case, this is actually a biotin-streptavidin interaction, which is pretty strong. It's easy to detect, admittedly. And so now we see a positive control, and then also a protein that's bound to our array. And we see not much background going on here. So this is our approach moving forward. We have procured over 200 metabolites, and we're starting to put them on and create a more complete small molecule endogenous metabolite microarray so that we can begin to probe things on a system scale. Importantly, though, and this is the caveat that's important for all high throughput screens, what you get is not always what you think it is. So, for example, first of all, we could have tons of false negatives. There might be things that don't actually bind strong enough for us to be able to detect it based on our washes here in the small molecule microarray, but they might actually bind. So there could be false negatives, but we have to give up on those. We can't concern ourselves too much with false negatives, though we can appropriately implement specific controls to make sure we minimize them. But we also could have false positives. Something could bind non-specifically to a protein, but not actually regulate it. Remember, that's what we're interested in figuring out, is what regulates these enzymes. And so in order to do this, we're actually doing a triage validation using mass spectrometry in an orthogonal approach. So what we do is we take our substrates and cofactors, add them to the protein with or without a candidate regulator, and then we do a mass spec assay to see, did my substrate concentration go down? Did my product concentration go up? How long did it take? And then figuring out how long it takes, both with and without the candidate regulator, we can figure out whether there's actually a kinetic impact of this regulator on our protein's activity. So this is our approach moving forward to make sure that we can validate and that everything that we get out actually is a regulatory interaction. So this is actually interesting because we now have a universal assay. And it's not like we're the first people to ever use mass spectrometry for enzyme kinetic assays. We're definitely not. But we're one of the few people who have realized that we can now look for essentially any arbitrary enzyme and use this approach to figure out if something is regulatory or not. Whereas typically people will look for a colorimetric assay to try and do kinetic measurements. We don't need to worry about getting specific colorimetric assays. We can use mass spec. So this project is in its early stages, but we're moving forward. And overall, we think that the workflow is going to be pretty successful. We've already gotten some promising results. And hopefully at the end of the day, we can get a library of regulatory interactions. And then what do we do with that? Well, we do metabolic engineering because that's our end goal. We want to be able to take all these interactions and say, well, now I can build a model of metabolism. Now I know what regulates what, and I can rationally engineer my cell to do exactly what I want it to in a metabolic context. Shifting gears. So we've looked at yeast. We've looked at trying to understand the regulation in yeast. We're also interested in the dynamics because that regulation specifically controls the dynamics. But here we're going to look in ovarian cancer. Now, there is more that we can learn from yeast metabolomics here. And that's because there's an interesting similarity between yeast cells and cancerous cells. These species up here and these species down here, the ones in black, they're what we call Crabtree positive, which means that they're respirofermentative. They prefer to make alcohol even in the presence of oxygen. Typically, you would expect yeast cells in the presence of oxygen to go through oxidative phosphorylation. But these guys still prefer to make alcohol even in the presence of oxygen. Interestingly enough, these guys up here, when you look at the metabolic profile and even at the transcriptional profiles, use totally different approaches to achieve this same macroscopic phenotype of being Crabtree positive. So what's going on there? How can we have essentially convergent evolution here that this is so important? This is something that's interesting and that we're pursuing. But then looking in a completely different direction, this respirofermentative approach is actually imperative in cancer. So if you look at normal differentiated tissue, in the presence of oxygen, it will go through oxidative phosphorylation and it'll give you tons of energy. In the absence of oxygen, it won't be able to go through oxphos, and it'll go through pyruvate and make you some lactate, and so you essentially have anaerobic glycolysis, which is less energetically efficient. This is what I was talking about, what we'd expect in yeast cells. However, when you look at proliferative tissue, or specifically in a tumor, you don't have this expected behavior. Instead, with or without oxygen, these cells choose to largely bypass oxidative phosphorylation and send tons of flux through anaerobic glycolysis. So this makes them necessarily respirofermentative. Even in the presence of oxygen, they still prefer to produce alcohol. So now we have a direct analogy between yeast and cancer, and this is sort of how I got pulled into it, because we have yeast as a model organism and we can study it that way, and then we can also go and study specifically cancer cells. And this respirofermentative approach has been recently acknowledged as a hallmark of cancer. One of the things that everybody is realizing is critical to cancer. One question that we could ask, so this is called the Warburg effect. I don't know if I didn't mention that yet, but this Warburg effect, we might wonder, is this actually somehow a critical cause, or is this maybe just a byproduct? Is this a necessary condition, or is it just something that happens anyway? And so people have done some experiments with dichloroacetate, which actually stimulates pyruvate dehydrogenase, which activates oxidative phosphorylation. Okay? So if we take our cells, we add DCA, we can then stimulate oxidative phosphorylation. So these cells that typically were not doing oxphos, now we can make them do oxphos. So we take our cancer cells and we make them be oxidative phosphorylation. We make them do oxidative phosphorylation. And what we see is a decrease in their growth and also an increase in the amount of apoptosis. Increased growth and decreased apoptosis are two hallmarks of cancer. So by decoupling this lack of oxidative phosphorylation from the cancerous phenotype, we've actually shown that we can decrease all of the other, or a number of other hallmarks of cancer. So this suggests to us that this Warburg effect is not just a byproduct of other things that are going on, that it's actually critical to cancer, and that it's critical for us to understand why and how it's occurring. Now, unfortunately, not a lot is known about cellular cancer metabolism. So I'm talking about it on an intracellular level, what the cells are doing with all these analytes. A lot of the current work that's out there is still very genetically focused. So even though, so cancer research for the past 20, 30 years has been extremely genetically focused. And then all of a sudden we remember something that was suggested 80 years ago, this Warburg effect, that metabolism might be important. But even though people are still trudging forward on the genetic aspects of metabolism and doing great work discovering things like that pyruvate kinase M2 isoform that I talked about, but people aren't yet looking on an intracellular level at metabolism. When people do look at metabolism, try to measure metabolites, what they're typically doing is measuring biomarkers in blood and other fluids. Now, this is still an absolutely critical thing to do, and I'm not discounting this in any way, shape, or form. But the biomarkers, the circulating metabolites in blood, there are way too many tissues that blood interacts with for us to be able to directly biologically interpret what the meaning of an upregulated alanine concentration in your blood is, right? So we don't know what this means in terms of a cellular level, which if you're looking to actually attack cancer metabolically, you need to understand what it's doing on a cellular level. And so by and large, the systems level cellular metabolic dynamics aren't well characterized, and so we would like to move towards that. And then I showed up at Georgia Tech, and I was looking to do some cancer work, and then I met John McDonald, and he pointed me in the direction of ovarian cancer, convinced me that this is a really important thing to study, and it is. Ovarian cancer is actually the 5th leading cause of cancer death in the United States. It has a 30% 5-year survival rate. So we're talking about when you're diagnosed with ovarian cancer, typically you're diagnosed kind of late, the odds aren't too good. And so getting an improvement in ovarian cancer treatment could be absolutely pivotal in improving these odds. And so that is our specific focus in collaboration with John McDonald here. So our goal is to characterize these metabolic dynamics in this model system. Specifically, we want to characterize them in response to in vitro perturbations, in vitro conditions that are characteristic of the conditions that the cells see in vivo. So if you think about what happens in a tumor, you're going to see at the center of the tumor there isn't going to be much glucose, there's going to be acidification, there's going to be hypoxia, and hopefully there's eventually going to be chemotherapeutics there. So we want to understand how do the cells respond in a time course profile to these changes in their condition, and how does that help us understand what we could do to specifically metabolically target them. Another thing that we're interested in this project, which I'm not going to go into today, is the differences between bulk tumor cells and cancer-initiating cells. There's actually strong evidence suggesting that there's a different metabolic phenotype between the general tumor cells and the chemo-resistant cancer-initiating cells that are actually one of the leading causes of metastases. So this could be useful for a uniquely targeted treatment towards these guys, but I won't get into that today. So our experimental workflow is kind of simple here. We take our cells, we apply some perturbation, we have some control unperturbed cells, and then we do time point sampling. Depending on the experiment, it could be anywhere between 2 and 6 days. Then we take our samples, we quench them because we want to stop cellular metabolism as soon as possible because it's actually metabolism is a fairly rapid process. And so if you let your cells sit there for a minute or two, the metabolite levels could drastically change. So we want to quench that metabolism as soon as possible, and then we derivatize, which means to functionalize all those molecules so that they can actually fly on our GCMS. And then we do our GCMS analysis and then do tons of data analysis to figure out what on earth just happened, because I now have biological triplicate plus a control, somewhere between 5 and 15 samples, and then I have hundreds to over 1,000 metabolites. So there's tons of information there that we need to be able to sift through, identify what's going on in there. Some of that data analysis is simple, some of that data analysis is not simple. But on a basic level, one thing that we can do is just do a sanity check, look at individual metabolite profiles, and see what's going on. Are there statistically significant changes? Or in this case, do they do what we expect them to do? So here what we actually have is in a glucose deprivation experiment, what does our extracellular glucose look like? Which you would expect, it would go down pretty quickly, it would all disappear, and that's actually what we see here in our experiment, and then our control doesn't show that behavior. So this is a quick and dirty check just to make sure that things are behaving like we want to, and then when we get an interesting hit in some sort of multivariate analysis, we can come back to these and start to try to interpret what's going on when I look at this time course profile. Another approach that we use is a dimensional reduction approach called principal components analysis. So what this does is it takes all of our variables and it tries to reduce them into 2 or maybe 3 variables. And the way that it does that is via a linear algebra approach. The important takeaway from this is to understand that principal component 1, 2, and 3, these axes represent axes of major variation. They're actually a linear combination. So a tenth of variable 1 plus half of variable 2 minus a tenth of variable 3. A big linear combination of all your variables that when put together represents the most variation in this principal component. It represents the most variation that you can get. And we can do these in successive orthogonal principal components. And what you've then done is reduced your hundreds to thousands of variables into 2. And then you can plot your data in this new data space and then do interesting things occur. And so if we see separation of alike data points, that will tell us that there's some biological relevance. So here we have things that are color-coded and also shape-coded. And we see that these purple guys are the same shape clustered together and away from these purple guys are the same shape. So this begins to suggest to us that there's something going on here. And we can go into more depth to try and figure out what these principal components are telling us. So hopefully we understand what we're looking for here are clusters of things that are the same color, the same shape, that are close together in our lower dimensional space. We also do metabolite set enrichment analysis, which is after we get all of our analyses, we figure out what's significant, what's not, and then we start to try and figure out what are the commonalities behind all this significance. This is really a meta-analysis level, and I'm not going to get into our work in this either. So we start, just like with our yeast experiment, we start with a basic glucose deprivation experiment. What happens when the cells run out of glucose in their medium? And this is something that, if you're looking at a tumor cell, in the middle of that cell, there's not going to be angiogenesis. Not a tumor cell, sorry. If you look at a tumor, there's not going to be angiogenesis to get you a lot of glucose into the middle of that tumor. So there's going to be a decreased glucose concentration. And so when we measure either extracellular or intracellular metabolites, we can just look at what's in the medium, or we can bust open the cells and look what's in them. What we begin to see here for our glucose deprivation experiment, and all of these are actually normalized to their control time points. So this is 15 minutes experimental condition normalized to the control at 15 minutes. So these are all changes. What we begin to see are a cluster of similar time points. So these guys are all clustered together. So there isn't necessarily tons that we can make out of that, because we see that the brown, red, purple, and blue, they're all kind of mixed together. But we then see two distinct clusters over here, this blue cluster and this green cluster over here. So this is telling us that in 24 and 48 hours, we begin to see substantial changes with respect to the rest of the time points, that maybe there aren't tons of early changes, but as we get into our culture, we can see substantial changes, and that there's significant change in between 24 and 48 hours. When we look at the intracellular, so now let's bust open the cells and measure what's in them, what we see again are our early time points here, which we don't see much change intracellularly in the first 15 minutes. But then, if you kind of squint, this is purple, blue, green, light blue. And we see purple, blue, green, light blue. So we see this time evolution here of going from earlier time points to later time points. So we're beginning to see, even in 2 or 3 dimensions, this clustering of multiple different time points in a profile that then gives us something that we can go into more detail to try and understand what are the loadings, what are the important variables in each of these principal components that are defining this variation. So we can see metabolically distinct changes, and it's kind of a gradual change with time, because you don't see things until, you don't see strong changes until 24 or 48 hours, but we can see changes. The next experiment that I'll tell you about is acidification. So again, as these cells are not undergoing oxidative phosphorylation, they're creating a lot of lactate. That's going to cause our pH to go down. And so we can take our cells growing in vitro and we can decrease their pH. And what we'll see here is, first of all, we can see long-term changes in the metabolite profiles. So here are the rest of our time points, and then here are our 24 and 48 hour time points. So we see long-term changes. Once you get to 24 to 48 hours, you could see substantial changes, and these are actually intracellular. I apologize for not labeling that. These are the intracellular metabolites. After 24 or 48 hours, metabolism has changed substantially. We also see, and we're pursuing this to see just how real this is. So you see these guys over here. This is a big cluster of 0, 30 minutes, and 8 hours. 15 minutes somehow seems to be showing this weird pulse, that it's actually more divergent from all these other time points. And so essentially, if you go from brown to red to purple, you go brown out to red and then back to purple. So we're not sure if there is some brief, quick response that the cells are showing to this change in pH. Or, I mean, it's possible this is an artifact, but we're going to be studying this in more detail to see if this is an initial pulse response that then allows a secondary pulse response or not. Overall, if you look at the gross metabolite profiles, we see 100 metabolites that have statistically significant dynamic profiles compared to control. So there's tons of interesting stuff going on here. We're only beginning to scratch the surface. Finally, the mesenchymal epithelial transition, MET. So this is a transition between cell types that also shows metabolic underpinning. So mesenchymal cells are typically more characterized with increased invasiveness, and it's believed that there undergoes in a tumor a transition from epithelial to mesenchymal that allows these cells to actually cause more metastases. So this is the super simplified 5 to 10 second explanation. But this is an important process in cancer recurrence here. And so we wonder if this change in phenotype of the cells actually exhibits any metabolic underpinnings. And what we find is that it actually does. So we did an experiment where the cells started off in a mesenchymal phenotype, and they were made to induce to an epithelial phenotype, and then they went back. So at 0 hours, we started inducing a change, and what we saw is that by 12 hours, we saw a change. And then things kind of relaxed back, because at 1:44 we expect everything to be back to kind of normal. And so we saw that by 24 hours things were kind of back to normal. And then at 48 hours it went out again and there was a big change. And then we came back to everything being kind of close to normal. So we see essentially two different metabolic pulses, one in this direction and one in this direction, otherwise characterized by normalcy. But again, the interesting thing here is that 24 hours is in between these two pulses and yet looks more like the normal mesenchymal phenotype. So we're not really sure what's going on here. I put that up again, I'm sorry. So we're not really sure what's going on here. Could this actually be real? Well, when we look at then the intracellular phenotypes, the intracellular metabolites, we actually see the same exact thing. We have our red guys here and we have our blue guys here that are away from the bulk cluster of everything else. So the fact that we see it in both intra and extracellular metabolites suggests that this might actually be something that's real. So I've shown you the overview of this. This is looking at all of the analytes at once. And now our task and what we're continuing to do is to dig in and figure out what are the specific metabolites that are causing these changes. So taken together, we see the importance of using metabolomics and looking at our metabolites, that we can see changes in profiles, we can see unusual double pulses, and we can see all these things that help us to characterize the metabolic dynamics and hopefully eventually the regulation of these cancer cells. So that wraps us up pretty much. We started off in yeast trying to understand how we could possibly get substantial changes in metabolism, even though transcription is sort of changing gradually. And we believe that a lot of that comes from the regulatory interactions, which we then believe substantially affects our metabolic dynamics. And one last thing before I go, I just want to point out that all these applications have been looking at very focused in vitro type studies. But we can also take this approach and do more in vivo type studies as well. So we actually have a collaboration with the USGS and the Georgia Aquarium, where they actually had bred and wild salmon. And we did metabolomics on their serum. And the amount of diversity that we expect in these wild salmon is significant. And yet, in two principal components, we're able to separate, with one exception, all the wild salmon from all the bred salmon in response to spawning. So we can do these basic biofluid analyses that show us, well, there's a substantial difference between these two classes, and then we can dig in and try to understand, in essentially the new frontiers of metabolomics, systems that people haven't even bothered to look at metabolism yet, which salmon is one of them. So we can apply this to then a number of different systems here. We have a very modular and widely applicable approach here. So I just wanted to point that out too. So that's about it. I, of course, want to thank all the people that actually did all the hands-on work in the lab. So Rob has done a ton of our data analysis, and Katie has done a lot of her own data analysis too. Katie generated all of our ovarian cancer data, and Mackenzie, with a little bit of help of Anna, who used to be bolded here, I don't know what happened there, has generated all the metabolite regulatory interaction data. And then, of course, we've had some great collaborators like John McDonald and Lijuan Wang, who's done a lot of the cell culture and helping Katie to get her feet. And then Al Dove from the Georgia Aquarium, who coordinated that salmon project, and then some generous funding sources. So with that, I'll take any questions if you have them. Yeah, Hong.
To identify the regulatory interactions, the way that you use, I was wondering, do you assemble different conformations of the metabolites on the array, or for each metabolite you have only one conformation, so the protein would be able to see only one conformation?
So the chemistry that binds the metabolite, so it's a covalent binding of the metabolite to the array before we probe with the protein, and it's actually it is chemical specific but not stereospecific. So there are a bunch of different functional groups that can actually catalyze or that can actually be the attachment point. And so if you have multiple of those chemical groups on your metabolites, which usually we do, then it's random which of those are actually the conformation. So this is a great point because if you have a metabolite, if we treat it as vaguely like my fist, the place where it actually binds the protein could be this functional group. And so if you have this functional group facing down on the array, then you wouldn't be able to detect that interaction. But luckily, we typically have multiple functional groups, and so we'll have a mixture of both conformations so that we do expect to decrease the number of false negatives. But that is one prominent source of potential false negatives. Hong had a question? Yeah.
Really nice talk, Mark. I was actually wondering, as you showed in the cancer story, what is I guess the diversity in cell origin as compared to the time differences that you observed? Or have you done different cell sources?
So we've, for now, just stuck with, we have one cancer cell line, OVCAR3, that we've been working with. So we haven't gotten into looking at cross-cell line differences just yet. From experience, we expect to see, if you were to take 2 or 3 different cell lines and just do the same basic culturing and then project on a PCA, you're going to be able to see differences. There are definitely metabolic differences. However, by experience and with no data to back this up, if you were to do some sort of substantial perturbation to the cells, like taking away their glucose, when you do the PCA projection, that is going to dominate any separation over the actual cell line. But we do expect to see a baseline difference, a small baseline difference in metabolism between cell lines. Yeah.
So particular, so when you were doing the heat map, showing a phylogeny as it compared to the genes, did you use a subset of the genes off of that chip?
So that picture, that specific picture that I showed you, was all of the genes. We then also, because we were comparing it to metabolism, right? So we then did a subset of all of the metabolism-related genes and still saw exactly the same behavior. And then we also bootstrapped and took however many metabolites that we had. I think it was 240. And then we took 240 random genes 100 times. And every time we always saw the same thing. So it wasn't just an artifact of sample size or the specific sample set.
Mark, can you just give us an overview or a sense for how the metabolites are actually identified in the mass spectra? Is there really a signature, or how accurate is that?
There is a signature. How unique the signature is not always so hot. So if you look at isoleucine and leucine, so we have 2 isomers, those guys are going to have fairly similar mass spectra. There are going to be differences, and depending on how complex your chromatogram is, so this is unfortunately where the separation actually plays a role in how good of a spectrum you can get. And so if you have good separation, then you can identify them fairly well. But if you're looking at things that are very close isomers, and definitely stereoisomers we don't have a chance at, but otherwise structural isomers, those can be kind of close, but distinguishable if you have a clean enough chromatogram. But otherwise, if you're looking at leucine versus any other amino acid, it's fairly noticeable. I mean, it's impossible to say that you have an absolutely unique mass spectrum, but they're pretty reliable as signatures. The other complicating thing, and we've actually had to invest a lot of time in this and effort, is that you can get a slightly different mass spectrum on our machine versus an Agilent quad versus an Orbitrap. You can get slightly different mass spectra. And so it's best if you actually generate your own library, and so we've invested some time in that too, to try and decrease these issues of, is the library mass spectrum, if I go to NIST, I can get a whole library of mass spectra, but is that actually what I should be seeing in my system? And so we've been trying to deal with that too. Yeah.
Is there a difference in fingerprint in vitro versus in vivo in the metabolomics? Because, you know, one of those is 2D versus the other 3D. One of those is like the variables are very like stable. The other ones are completely unstable.
I agree. So At the end of the day, when it comes time for you to develop a drug that is specifically metabolically targeted, you need to have much better in vivo studies. I 100% agree that in vitro is going to be a different beast than in vivo. What we're limited by is obviously it's difficult to get any sort of time course dynamic sampling in vivo. And the reason why we need that time course sampling is so that we understand the dynamics and the regulation. If you take one single snapshot in time, that might give you a different perception about what's going on in the cell than what it's actually doing, right? Because if you took a different snapshot, you might have gotten a different picture. And so by having multiple snapshots, we can then in vitro begin to identify what are the things that we should be targeting in metabolism, and then find ways to target them and then try it in vivo. But I agree that in vitro is going to be different than in vivo, and this is one of those things that we just have to bite the bullet and do the experiments in the model system that we have and cross our fingers that it translates well to in vivo. But we also are working on developing approaches to better model an in vivo system. So Katie also has as part of her project that she actually wants to have tumor stroma interactions. And so moving towards having that is more reflective of in vivo, though still not quite there. Yeah, Brandon.
You might have already answered my question, but could you, in a sense, meet in the middle of, like, what if you do tumor fibroblasts, for example, cold cultures? Would there be evidence that there would be actual interactions between those two, manipulation between those two via metabolites?
I would say absolutely. If you look, so here's where my mammalian cell biology begins to falter. But if you look definitely in the brain and also in skin, you'll see that there are specific cell types that are actually more metabolically active that then provide metabolites as energy sources to other cell types that are less metabolically active. So you would definitely see, given the right model system, changes of isolated versus co-culture. And I'll say I don't think that there's tons of work that's been done there. All right. If there are no other questions, thank you so much for your time and for coming out, and hope you enjoyed it.